Hi guys and welcome to my channel. Myself Dr. Akshay, and in today's session I'm going to be discussing about the topic the structure of microcirculation and capillary system. If you guys want to learn more about the circulatory system, then click subscribe and the bell icon. So let's do this together. Coming to the topic structure of microcirculation and capillary system. Here, arteries should branch 6 to 8 times for becoming arterioles. And these arterioles will always have a diameter of 10 to 15 micrometers. These arterioles will then branch 2 to 5 times in order to become capillaries. These capillaries will always have a diameter of 4 to 9 micrometers. So, see this figure. You can see this is the arteriole. As you can see from this figure, this arteriole is highly mascular. These are the mascular structures which are attached towards this arteriole. These are the mascular structures. And because of these mascular structures which are attached towards this arteriole, these can increase and decrease its size manyfold. I mean, by increasing and decreasing its diameters. Another structure which is attached towards this arteriole is meta-arteriole. In meta-arteriole, this meta-arteriole does not have continuous mascular structures which are present in this arteriole. Whereas in meta-arteriole, there are smooth muscle fibers or smooth muscle structures which are attached at intermittent points. You can see these are the smooth muscular structures which are attached at intermittent points. And next you can see another structure. They are precapillary spinchters. So these are the precapillary spinchters. From here, capillaries will arise. You can see capillaries, they are arising from these precapillary spinchters. And these precapillary spinchters are composed of smooth muscular structures. These smooth muscular structures are the same muscles or the same smooth muscles which are present in this meta-arteriole. And these precapillary spinchters can either close or open towards the entry to the capillary. And you can see another structure, and that is the venule. So this is the venule. And these venules are larger in diameter while comparing with this arteriole. And also these venules are having lower muscular density while comparing with this arteriole. And also the pressure in the venules are very lesser while comparing with these arterioles. This is because in arterioles the lumen is very narrow, whereas in venules the lumen is very large. They are having larger diameter. So because of this narrow lumen, the blood pressure in arteriole will be very high while comparing with these venules. So next, coming to the structure of capillary wall. So see this figure. In this figure you can see these are the endothelial cells. So this is one endothelial cell, this is another endothelial cell, and this is another endothelial cell. And also you can see these endothelial cells are adjacent to each other, and in between each endothelial cells there's a gap. In between these two endothelial cells there's one gap. Again, in between these two endothelial cells there's another gap, and these gaps are known as intercellular clefts. And these intercellular clefts plays a major role in the diffusion of soluble substances through the capillary membrane. So we came to know that soluble substances diffuse through these intercellular clefts. So what about macromolecules? How macromolecules can be transported through this capillary membrane? Macromolecules are transported across the capillary membrane through some small invaginations which are present over this endothelial cell. You can see many small invaginations. And these small invaginations are known as caveola. This caveola contains caveolin. Caveolin, they are protein structures, and these protein structures will then polymerize with these sphingolipids, phospholipids, and cholesterol molecules to form caveolae. So note one important point, caveola are the invaginations which are present in this endothelial cell, whereas caveolae that are formed from caveola plus caveolin. So the wall of this capillary is composed of a single layer of endothelial cells. And this single layer of endothelial cells are covered by a basement membrane. And this basement membrane is situated just outside the capillary wall. The internal diameter of a capillary is almost 4 to 9 micrometers. This is barely enough for RBCs and other blood cells to squeeze through it. The rates of thermal motion of these water molecules and water-soluble ions and molecules are so rapid. And this makes these substances to diffuse with ease between the interior and exterior of the capillary wall, that too between these intercellular clefts. The pores in the capillaries of some organs will have special characteristics to meet the peculiar needs of the organs. In the brain, the gap between the adjacent endothelial cells are so tight. This allows for only small amount of oxygen, carbon dioxide, and water molecules to pass through them in a little by little amount. Whereas in the liver, the gaps between adjacent endothelial cells are so wide, so that all dissolved substances of plasma, including plasma proteins, can enter from the blood into the liver tissues. And next, coming to the pores which are present in the gastrointestinal tract. The pores which are present in the gastrointestinal tract are always in a midway between that of the muscles and that of the liver. In the glomerular capillaries of kidney, numerous small oval windows are present. And these numerous small oval windows are known as fenestrae. And these fenestrae penetrate all the way through the middle of the endothelial cells. And this helps for the filtration of tremendous amounts of ionic substances and molecular substances through these glomeruli. So that covers the topic structure of microcirculation and capillary system. I hope you guys learned about this particular topic. Thank you so much, guys. See you guys in the next session.